Today we turn our attention to human embryonic stem cells. Human embryonic stem cells can divide without limit, and yet they can maintain the potential to make all the cells of the body, a property known as pluripotency. This remarkable potential makes them useful for basic research on the function of the human body, for research on drug discovery and testing, and as a source for cells and tissues for transplantation medicine in the field of tissue engineering and regenerative medicine. In 1998, James Alexander Thomson was credited for the first successful isolation of human embryonic stem cells. He published his findings in the journal Science in 1998 under the title Embryonic Stem Cell Lines Derived from Human Blastocysts. The procedure for isolating human embryonic stem cells is called immunosurgery. It is done using a dissection scope equipped with a heated stage. And in this procedure, cells of the trophoectoderm are destroyed by brief exposure to antibodies directed against human cells in tandem with complement activity. Here's a brief video demonstrating the concept of immunosurgery.
Embryos at all different stages of development, and we culture them in vitro until they reach the proper expanded blastocyst stage. These are examples of how the embryo develops from the first, the one cell stage, to the early blastocyst, and then the expanded blastocyst, from which we derive human embryonic stem cells. High-quality embryos chosen for derivation are then transferred through three drops of acid thyroids, where you'll leave it and watch for dissolution of the zona lucida. The immunosurgery process is a non-specific process where the rabbit anti-human antibody will bind to the outer layer of cells, in this case is the trophectoderm. Upon application of guinea pig serocomplement, all of these cells with antibody attached will be lysed.
So as you can see, this leaves behind the cells of the inner cell mass, which are the source of human embryonic stem cells, which can be divided and basically cultured on mouse embryonic fibroblast feeder monolayers to allow them to grow under the proper condition and to differentiate when induced to do so. For more information on the isolation procedure, you can follow the link to the Journal of Visualized Experiments that is shown below the video clip. By necessity, the ability to isolate and culture human embryonic stem cells was first described by using inactivated mouse embryonic fibroblasts as feeder cells, as well as fetal bovine serum containing culture medium. The problem is that these are xenogenic materials, and in 2005 there was a published report in the journal Nature Medicine that stated that human embryonic stem cells available for federally funded research are contaminated with non-human molecules from the culture medium used to grow the cells. This has led to a series of developments in the culture technology to basically move away from the use of xenogenic material. For example, the fetal bovine serum, which is a very non-defined biological tissue in a way, was replaced with knockout serum replacement, as well as FGF2. The knockout serum replacement is basically a defined serum-free formulation that has been optimized to grow and maintain undifferentiated embryonic stem cells in culture. These feeder-free culture systems enable culturing the cells on matrix-coated plates such as Matrigel. But Matrigel in itself is also a xenogenic material. Recently, human embryonic stem cell culture conditions have moved towards feeder-free cultures in chemically defined conditions, and these methods basically ensure that no xenogenic material is used in the culture condition, such that these cells can be used with confidence in the human patients eventually. One of the other concerns in embryonic stem cell technology is whether or not these cells that are destined to divide indefinitely might experience some abnormalities in their chromosomes. Therefore, these cells, when they're isolated, are typically subjected to karyotype analysis, which is a technique where chromosomes can be visualized under a microscope. Briefly, this technique involves allowing the cells to divide and then arresting them at the metaphase when the chromosomes are condensed and therefore visible. The chromosomes are then stained with dyes that show a pattern of light and dark bands. This is called the banding pattern. And these bands reflect differences in the amounts of A and T versus G and C nucleotides. The banding pattern for each chromosome is very specific and is consistent, which allows identification of each of the 24 chromosomes. Now, karyotype analysis can detect large chromosomal abnormalities such as loss or gain of an entire chromosome or portions of a chromosome and translocations. However, only big changes can be visualized by this type of analysis. And many of the changes in our genetic material that can cause disease are very small and require other methods of analysis to be detected. In addition to validating the integrity of the DNA by karyotype analysis, the embryonic stem cells must also be validated for pluripotency and their ability to differentiate in vitro. Typically speaking, these cells are allowed to grow in culture, and when they have grown enough, they can be split into many passages to grow sufficient numbers of cells, and then induced to differentiate. And their ability to differentiate into different phenotypes is basically judged by looking at the morphology of the differentiated cells, as well as certain features such as a contracting or a spontaneously contracting cardiac muscle cells. Additionally, one can also stain with antibodies that bind to specific cell markers that would identify a certain cell phenotype. This is called immunostaining or immunohistochemistry. In addition, the pluripotency of embryonic stem cells needs to be judged in vivo, and this can be done in a couple of different ways. The first method involves injection of these embryonic stem cells into ectopic location under the skin of an immunodeficient mouse, and then watching for these cells to grow a benign tumor called a teratoma. This teratoma is basically composed of advanced tissue types of all of the 3 germ layers, and these can be identified by immunohistochemistry as well as by morphology. The second method, which is the most robust method of demonstrating the pluripotency of embryonic stem cells, or any stem cell for that matter, is the ability of these cells to engraft into a blastocyst to eventually form a chimeric mouse composed of cells that have been injected into the blastocyst as well as the original host cells of the blastocyst itself. Experiments to demonstrate this typically involve embryonic stem cells from a reporter mouse, such as a GFP-positive blastocyst. These are typically injected into the cavity of a wild-type blastocyst and then transferred to the uterus of a pseudo-pregnant wild-type mouse. If the cells that are injected into the blastocyst, which we know are GFP positive, if they are truly embryonic stem cells and have pluripotency, then the offspring are chimeric mice of which all tissues are composed partly of host cells and partly of those donor GFP positive cells. And these could be identified using immunofluorescence as well as fluorescence in general to basically delineate the amount of engraftment of these embryonic stem cells and their ability to differentiate into the different cell types and tissue types. So now we start discussing the therapeutic potential of human embryonic stem cells. These cells can be used in a variety of different ways, and the first obvious application of embryonic stem cells is that which involves replacing or repairing tissue after damage or loss. To demonstrate a good preclinical application of stem cells in direct repair of damaged tissue, I'd like to present this paper, which was published in the journal Science in 2004, in which cardiac defects resulting from a specific mutation or knockout of certain genes results in embryonic lethality because of a heart defect. Specifically, this is a situation in which mice were generated with double or triple knockout of certain genes known as ID1, ID2, and ID3. These genes are acronyms for inhibitor of DNA binding differentiation proteins. And they comprise a family of proteins that heterodimerize with basic helix loop helix transcription factors to inhibit DNA binding of BHLH proteins. These ID proteins, in a sense, function to prevent premature differentiation of stem cells. While individual knockouts of these genes, ID1, ID2, or ID3 knockout embryos, are not embryonic lethal, the ablation of 2 of these genes or 3 of these genes, all 3 of them, leads to embryonic lethality. The triple ID knockout embryos die at mid-gestation due to a marked thinning of the myocardial wall, which results in death. Because of their pluripotency, and because they are touted for their therapeutic potential, it was hypothesized that embryonic stem cells can potentially correct that cardiac defect. To demonstrate whether or not this is true, experiments were done in which a small number of wild-type embryonic stem cells that are carrying a reporter marker, LAG-Z, were injected into a blastocyst of an ID mutant mouse. And this resulted in about a 20% chimerism of the heart tissue and complete rescue of the ID mutant phenotype. The chimerism was basically verified by the presence of blue cells, which basically is an immunostaining that reads the presence of LAG-Z expressing cells from the injected embryonic stem cells. In addition to the engraftment of these embryonic stem cells, it was identified in these experiments that it was not necessarily the direct differentiation of these embryonic stem cells into the heart muscle that resulted in the rescue of the mutant mouse. Rather, 2 signaling molecules secreted by these engrafted ES cells, Wnt5A and IGF-1, were basically identified as potential candidates involved in this rescue process. So, this suggests that the rescue effect is not subject to the differentiation of ES cells into the cardiac cell lineage that are normally associated with heart regeneration. Rather, the therapeutic effect of these transplanted ES cells depends on their secretion of defined factors that act either locally or at a distance via maternal circulation to trigger fetal myocyte proliferation in utero. So with that in mind, the next experiment was designed to basically demonstrate that embryonic stem cells therapeutic effect is really mediated by their ability to secrete factors that can have long-range signaling effects and that could be able to cross the placenta barrier to potentially rescue the developing heart. So to demonstrate this, IP injections of wild-type embryonic stem cells into female mice that are double mutant in ID genes were performed prior to conception with males of also a double knockout phenotype. This injection of these ES cells also resulted in partial rescue of the cardiac phenotype without incorporation of the embryonic stem cells into the fetal heart tissue. Interestingly, similar experiments were done, but instead of IP injections of embryonic stem cells, IP injections of IGF-1 were also attempted and resulted in similar rescue of the mutant phenotype. And this therefore demonstrates that the rescue of these defects by embryonic stem cells does not necessarily depend on engraftment of these cells into the tissue of interest or differentiation of these embryonic stem cells into that specific tissue type, but rather on their ability to secrete factors that could correct a defect in a specific genetic disease. In that sense, you can think of embryonic stem cells not only as regenerative, but also as medicinal factories of some sort. In the previous example, we saw a practical application of the use of human, or rather embryonic stem cells in their naive pluripotent state. As an alternative, embryonic stem cells can be expanded and then differentiated into a cell lineage of interest that then can be used to rescue a defect. One example of such an approach involves the correction of Parkinson's disease. This is a study that was published in our own Steve Goldman's neurology laboratory, in which they attempted to correct Parkinson's disease in a rat model of this disease. Parkinson's is a chronic neurodegenerative condition that does not have a cure, and it is caused by the death of dopamine-secreting neurons that reside in the substantia nigra part of the midbrain. These neurons project to the striatum, a brain region involved in planning and executing movement. Therefore, the loss of dopaminergic innervation leads to motor symptoms such as tremors, rigidity, and unstable posture. Pharmacological treatments and deep brain stimulation have side effects and bring only short-term relief of the symptoms, and this in general is a progressively degenerative disease. So, to address this problem, the researchers in Steve Goldman's lab developed protocols for the derivation of highly enriched populations of dopaminergic neurons, the type of nerve cells that will deteriorate in Parkinson's patients. And this protocol involved co-culturing these human embryonic stem cells with support cells of non-human origin before grafting into the adult animal. Now, the problem with this traditional approach is that the value of such derivation procedure for these human therapies is limited because of the presence of xenogenic material in cell grafts, which might cause problems in the patients. So alternatively, what Steve Goldman and his group did was co-culture these human embryonic stem cell derived neural precursors with immortalized human fetal midbrain astrocytes, and they therefore were able to achieve robust differentiation of these embryonic stem cells into dopamine producing neurons while avoiding the customary exposure of these human cells to xenogenic feeder cells. Indeed, when these cells were transplanted into a rat model of Parkinson's, this led to the integration of these human embryonic stem cells into dopaminergic neurons into the host brain and led to an amelioration of the disease symptoms. These were extremely positive results. However, they were confounded by the troubling finding that the inner cores of these grafts contained slowly dividing neuroepithelial cells that were tumorigenic. This basically is the nightmare that is associated with embryonic stem cells, the long-standing fear that stem cell therapies could lead to cancer. So, as you can see, there is a great potential for human embryonic stem cells for therapeutic applications in tissue engineering and regenerative medicine. And this hope is real, but there's also quite a bit of hype about what we can do with these cells. And I think that we are still taking our first steps towards being able to realize the potential applicability of these cells in the clinic. In fact, if you go to a very useful website, which is clinicaltrials.gov, you can search for ongoing clinical efforts that involve a variety of therapies. So if you use the keyword embryonic stem cells, you will get a listing of current clinical trials that involve the use of embryonic stem cells either in diagnostic applications or in interventional applications. And this is a screenshot of a search I did yesterday, basically showing that there are 31 studies found for embryonic stem cells that are current. So with this kind of information, you should appreciate that the promise of embryonic stem cells in therapy, in human therapy, is very real and being pursued very seriously. As a side note, as you begin to work on your term project or proposals, this particular search tool, the clinicaltrials.gov, could be a useful resource for you to try and include some relevant clinical applications of the topic you're researching. Now, in spite of their great medical potential, human embryonic stem cells generated enormous controversy because their derivation typically involved the destruction of a human embryo. This controversy is not settled yet and will probably never be settled, and had real implications on the debate, not only in science but also in ethics, religion, and political circles. And this has real ramifications on the availability of federal funding for this type of research, which we continue to have to deal with. So with these concerns about the ethics and politics of the use of embryonic stem cells and their real ramifications on the availability of funding for research and clinical development, the search has been on for alternatives to isolate cells that would have features similar to embryonic stem cells. And one of the earliest technologies that enabled such a possibility was the process of cloning, or somatic cell nuclear transfer. In this procedure, an egg cell nucleus is basically removed and discarded, leaving behind an enucleated egg. At the same time, a nucleus of a somatic cell, such as a skin cell, is removed and basically then introduced into the enucleated or deprogrammed egg cell. And once it is inserted into the egg, the somatic cell nucleus is reprogrammed by the host egg cell, and the egg which now contains the somatic cell's nucleus and the DNA material of the host of the donor cell, is basically stimulated with a shock to begin to divide. And after many mitotic divisions, this single cell forms a blastocyst, which can then basically be used to isolate cells from the inner cell mass. These cells have basically about 100% of the DNA of the original organism or the original donor, but they also contain maternal DNA from the host egg. The technique of transferring the nucleus from a somatic cell into an egg that produced Dolly, the sheep. This was an extension of experiments that had been ongoing for over 40 years before Dolly was generated. In the simplest terms, this technique used to produce Dolly the sheep, somatic cell nuclear transfer cloning, involves removing the nucleus of an egg and replacing it with the diploid nucleus of a somatic cell. This procedure is not straightforward. There are many limitations. First, there are stresses that are placed on both the egg cell and the introduced nucleus. These could lead to a high loss in resulting cells. For example, when Dolly the sheep was born, there had been 277 eggs that were used for cloning, which created only 29 viable embryos. Only 3 of those embryos survived until birth, and only 1, that is Dolly, survived to adulthood. So the procedure currently cannot be automated. It is basically marred with complications and inconsistencies, and would have to be done manually under a microscope. It is very resource intensive, and the chemistry involved in reprogramming this differentiated somatic cell nucleus and activating the recipient egg to divide to become a blastocyst is really far from understood at this point. This here is a video demonstrating the process of somatic cell nuclear transfer. So you can see a micropipette poking through the zona pellucida and through the egg to eventually suck out the nucleus and discard it. This now leaves behind the enucleated egg. And you can see here the pipette is basically spewing out the nucleus of the egg. And you'll see now that this enucleated egg is basically moved aside to obtain another cell. Now the next step, once the cell or the egg has been enucleated, is to use a pipette to also introduce the nucleus of the somatic cell. So here again, the pipette is going to poke through the membrane, zona pellucida, into the egg, and you can see very slowly the nucleus of a somatic cell being injected into the enucleated, deprogrammed egg. And so on. The procedure can be repeated for other cells. So the technology in which somatic cell nuclear transfer is enabled is not without controversy. The idea that this technique basically avoids the destruction of an embryo is in some sense true, but at the same time is still under the subject of concern about the use of an entity that is embryo-like, even though it's artificially generated. The other concern is that this technique can actually be used to clone whole organisms. Case in point, the sheep Dolly. And more importantly, concern arises that this technique with more advancements and refinements can eventually lead to human cloning. These concerns notwithstanding, stem cells that are generated from somatic cell nuclear transfer or cloning have real potential value. The first potential application involves the use of stem cell lines derived from cloning that can be made from a person's own tissue. And this has the advantage of being able to avoid immune rejection. In some sense, we're creating patient-specific stem cells. This is a schematic representation of this idea in which a skin cell, for example, as a source of nucleus and DNA, is injected in an enucleated egg to potentially generate different tissue types from embryonic stem cells that carry the same genetic material as the donor, which in a sense will also be the host for whatever tissue these cells are eventually programmed to differentiate into and then used to repair or replace a damaged tissue or organ. In addition to the direct therapeutic value that this option offers, one can consider a scenario in which a patient with a genetic disease can offer opportunities to generate cells that could be used in biomedical research. Here is an example of such a scenario in which a patient with amyotrophic lateral sclerosis, or ALS, also referred to as Lou Gehrig's disease or motor neuron disease, can provide material for research into the etiology of this disease, as well as to generate cells that could be used for drug screening for potential therapeutics that could alter the course of the disease or modify it. This is a real scientific need, and this technology, somatic cell nuclear transfer, really enables the possibility of using human cell-based models of the disease rather than the use of animal models, which often carry a lot of limitations that limit the ability to interpret the findings associated with them. A fourth possibility that offers some therapeutic value in the use of embryonic stem cells involves genetic alteration of these stem cell lines so that they can be implanted to act as local factories producing therapeutic molecules. And the concept is very real. In contrast to traditional gene delivery, which involves direct delivery of plasmids of naked DNA or viral vectors that carry certain gene sequences into the body, the idea here is to genetically modify embryonic stem cells and then allowing them to be differentiated or implanted into the patient to correct a particular defect. In contrast to the study that we presented a few slides ago by Steve Goldman, in which they have used naive embryonic stem cells that have been coaxed to differentiate by co-culturing with other human cells and other factors. This here is an example of a study that involves the possibility of using embryonic stem cells as a carrier of therapeutic genes. These cells were transfected with cDNA coding for tyrosine hydroxylase and GTP cyclohydrolase 1. These were then basically, when successfully transfected, were basically found to produce significant amount of IDOPA spontaneously in these stem cells. And when these stem cells were basically implanted into the striatum of rat brains, were able to demonstrate that they relieve many of the symptoms of Parkinson's that these rats have been induced to have. So this demonstrates the potential to use this genetic approach to modify stem cells to produce factors that could potentially correct a disease such as Parkinson's. So all of these therapeutic potentials and therapeutic applications of embryonic stem cells were really made possible by the development of this technique called cloning or somatic cell nuclear transfer. And while James Thomas is credited with the first successful human isolation of embryonic stem cells, the idea, and this was fairly recently in the 1990s, the idea of cloning goes back in history at least for 3 or 4 decades. In 1962, John Gordon discovered that the specialization of cells is reversible. He performed a classic experiment in which he replaced the immature cell nucleus of an egg cell of a frog with the nucleus from a mature intestinal cell. This modified egg cell developed into a normal tadpole. The DNA of the mature cell still had the information needed to develop all the cells of the frog. Therefore, it is Gordon's research that really taught us that the nucleus of a mature specialized cell can be returned to an immature pluripotent state. But his experiment involved the removal of a cell nucleus with the eventual introduction of the nucleus into other cells. So the question is, would it ever be possible to turn an intact cell back into a pluripotent stem cell? Notice the subtle difference in the approaches. In Gordon's experiment, a nucleus has been implanted into another cell to generate blastocysts from which cells that have ES features can be isolated. The question now is, can you take a somatic cell intact and reverse program it into a pluripotent stem cell state? And this indeed has been made possible by a recent discovery in 2006 by Shinya Yamanaka, in which he demonstrated that you can take intact mature cells from mice, and these could be cells from skin, for example, and these cells can be reprogrammed to become immature stem cells. This is simply done by introducing only a few genes, and one could reprogram the mature cell to become a pluripotent stem cell, which has all the features of embryonic stem cells and have the ability to develop into all tissue types in the body. So these two seminal experiments and their important findings that led to revolutionary ideas in terms of the ability to use stem cells in research, which have been separated by some 40 years, have eventually been awarded the Nobel Prize in Physiology and Medicine in 2012 for both Gordon and Yamanaka. Gordon, in this case, was recognized for the experiments that eventually laid the foundation for cloning or somatic cell nuclear transfer, while Yamanaka has been recognized for discovering the key genes that can be used to induce pluripotent stem cell states in somatic cells, technology known as induced pluripotent stem cells or iPS cells. So let's spend the next few slides discussing the science behind the technology of iPS cells. This is a classic experiment by Yamanaka, which was published in the journal Cell in 2006, and this work is basically the foundation for which he was honored with the Nobel Prize. The approach to basically understand how you can take a somatic cell and reverse it into a pluripotent stem cell state involved a clever experiment in which selective reverse engineering of the cell was used. What Yamanaka and his group did was develop an assay in which the induction of pluripotent stem cell state can be detected by basically endowing these cells that have been successfully de-differentiated with the ability to resist strong antibiotics such as G418, which is an analog of neomycin. And to do this, they basically had to introduce a fusion cassette that contains neomycin-resistant genes. And this cassette had to be fused with a specific gene that would be expressed in the mouse embryonic stem cells and early embryos, but is then silenced in more mature adult somatic cells. And one such gene is called FBX15, which is specifically expressed in the mouse embryonic stem cells and in the early embryo, but is dispensable during normal development. So somatic cells from the rat tail, from mouse tail, were basically isolated, and these are called adult fibroblasts. And alternatively, mouse embryonic fibroblasts were also used, in which these fusion cassettes that contain the FBX15 gene as well as the beta G geocassette were stably introduced. And then the experiment involved introducing about 24 candidate genes that were thought to play a role in pluripotency. So the idea that if the cells, when they're transduced with these 24 candidate genes, if they are able to be reversed completely into a pluripotent state that expresses FBX15, then these cells would be highly resistant to neomycin. And if they fail to de-differentiate into this embryonic stem cell state, then they would be very sensitive to neomycin and similar drugs. So this then allowed the cells that have been able to be de-differentiated into a stem cell state to be selectively enriched with the treatment of neomycin or G418. And you can see in the panel in the bottom that in the case of introducing all of the 24 factors that have been identified, numerous black dots, these are basically clonal populations of these cells that have the ability to resist neomycin and therefore express that very early embryonic gene of FBX15. And this basically suggests that these cells that survived the neomycin treatment are indeed pluripotent stem cell-like cells. So next, to assess which of the 24 candidate genes is dispensable for this process, an experiment was done to basically allow for sequential withdrawal of these genes one at a time, and then evaluating the potential for colony formation with each gene withdrawal. So as you can see in the yellow highlighted box, when you introduce all 24 candidate genes, you can see that colonies have been formed by as early as 10 days and continue to form even at 16 days, suggesting that these cells do indeed have resistance to neomycin, and by corollary, they have the FBX gene expression, which is sort of the marker of the pluripotent de-differentiation. Interestingly, when you introduce only one factor, none of these 24 factors induce G418 resistance, suggesting that none of these factors is sufficient to induce a pluripotent state differentiation or de-differentiation. And when they basically performed the experiment by withdrawing one factor at a time, they examined the situations in which no colonies formed at day 10. And this gave them an indication that these candidate genes that have been withdrawn in these scenarios are indispensable for the dedifferentiation into a pluripotent state. So this reduced the matrix from 24 candidate genes down to 10 genes. And the experiment was then repeated with the introduction of these 10 factors and the selective withdrawal of one at a time, which then identified further that a subset of 4 genes is absolutely necessary and sufficient to induce these somatic cells to become pluripotent cells. And these factors that have been identified for IBS cell formation are OCT3/4, KLF4, SOX2, and c-MYC. And indeed, the cells that have been generated from a skin cell or from a fibroblast have the hallmarks of embryonic stem cells and their ability to form embryonal bodies, as you can see in the figures at the bottom. And this is very distinct from the morphology of these mouse embryonic fibroblasts that have not been transduced or induced to become pluripotent cells. And this is the image on the far right at the bottom. So this clearly demonstrates that this IPS technique can actually induce the somatic cells to become pluripotent stem cell-like organisms. So to reiterate, no combination of two factors could induce the formation of pluripotent stem cells. And if you used a combination of 3 factors only, there were scenarios in which small colonies formed, but these could not be maintained in culture. The combination of OCT3/4, KLF4, SOX2 without c-MYC did indeed result in the formation of some 36 G418 resistant colonies. However, these colonies exhibited a flat, non-ES cell-like morphology. The combination of OCT3/4, KLF4, and c-MYC without SOX2 did in fact lead to the formation of 54 G418 resistant colonies. The morphology of these cells indicated that this combination can in fact activate FBX15 locus, but the change induced by these factors alone was different from all 4 genes, in that they did not necessarily have the induced pluripotent stem cell state. So having now identified those factors that are necessary and sufficient to take mouse embryonic fibroblasts or adult fibroblasts and turn the clock back to turn them into induced pluripotent stem cells, the Yamanaka group went on to demonstrate that these cells indeed have embryonic stem cell-like features, and demonstrated that when you look at their ability to self-replicate, these cells continue to divide almost indefinitely in culture without slowing down, whereas the mouse embryonic fibroblasts from which they were derived undergo maybe less than 40 days of passaging, potentially reaching that half-life limits that you might recall being discussed in the cell culture classes. So this basically demonstrates that these induced pluripotent stem cells have the ability to self-replicate indefinitely. Then they looked at a pattern of gene expression that has been shown to be robust in these embryonic stem cells, and these experiments suggested that depending on the clone that you look at, there may be some key omissions in the expression of certain factors. However, one factor in particular that has been typically used to characterize embryonic stem cells is nanog, and in nanog or in embryonic stem cells as well as uniformly in induced pluripotent stem cells, nanog expression was maintained. Next, Yamanaka and his group basically looked at the epigenetic changes to these iPS cells that were generated from mouse embryonic fibroblasts, and they looked at a couple of different clones for this purpose, and using some genomic sequences of the promoter region of OCT4, nanog, and FBX15, they basically did an analysis of histone acetylation as well as CpG dinucleotide methylation. And these experiments demonstrated that these iPS cell lines that are derived from mouse embryonic fibroblasts are similar in some sense, but they're not identical to embryonic stem cells. Okay, so if you look, for example, on the figures, the bar charts on the top right hand of the slide, you will notice that the data indicates that the promoters of OCT4 and nanog had increased acetylation of histone H3 compared to the somatic cells, but also decreased dimethylation of lysine 9 of the histone H3. Okay, and if you look at an analysis of the methylation patterns, which is these dot plots in panel F, these basically suggested that CpG dinucleotides in these promoter remain partially methylated in iPS cells and not completely unmethylated. In addition, these clones of iPS cells were positive for high activity of alkaline phosphatase, stage-specific embryonic antigen 1, which is expressed at the morula stage in a mouse embryo, and these cells have a high telomerase activity. So these are basically indications that these iPS cells share some features of embryonic stem cells, but they're not identical in terms of their epigenetics. Finally, the pluripotency of these iPS cells that are derived from mouse embryonic fibroblasts was also investigated in vitro, and this was done by allowing these cells to form embroid bodies and then differentiate on their own. And this was confirmed by immunostaining as well as morphological analysis. And these studies demonstrated that these iPS cells can indeed differentiate into all three germ layers. And the panel of immunostaining shown in color in the bottom here demonstrate that these iPS cells were able to generate cells of the smooth muscle phenotype, the liver phenotype, as well as the neurologic or neuronal lineage. In addition, when these iPS MEF cells were injected into blastocysts of wild-type animals, they were able to demonstrate that they engraft into the chimeric animal and contribute to the formation of various tissue types that belong to the three germ layers. And this was again confirmed by immunostaining of different tissues that were harvested from the animals that resulted. So these previous experiments were done with a mouse embryonic fibroblast, and the big question, and where a lot of the excitement really resides, is whether or not you can take an adult fibroblast, right, and then program it or reprogram it such that it becomes an iPS cell. And to demonstrate this, Yamanaka and his group took tail-tip fibroblasts from an adult mouse and repeated pretty much the same experiments, demonstrating that yes, with the introduction of these factors, these four factors, you can reverse these somatic cells that are fibroblasts to an iPS state, and that these cells have the ability to differentiate into different tissue types across the 3 germ layers. Again, demonstrating that these iPS cells that have now been derived from adult fibroblasts or adult somatic cells are indeed embryonic stem cells in their nature. A more robust test that involves the assessment of the contribution of these iPS cells to the formation of a chimeric embryo does demonstrate that these cells do indeed engraft into the embryo and contribute to the formation of different tissue types like the heart, liver, neural tube, skin, gonad, and the GI tract. So, taken together, this was a very exciting experiment and a very exciting discovery of a new technique that would allow us to create embryonic stem cells from somatic cells. This was done in the mouse, and after that it was demonstrated that you can actually generate human iPS cells with the introduction of a similar set of factors, including OX4, SOX2, and NANOG, but instead of c-MYC, which is an oncogene, and there was concern about introducing a gene that would potentially cause cancer, some other combinations of factors that include NANOG and Lin28 seem to demonstrate reproducible conversion of somatic cells of human origin to an iPS state. So we are just about out of time at this point. I'm going to stop here, and we're going to pick it up on Monday in class to discuss potential uses of iPS cells in the clinic, as well as discuss other types of stem cells that are derived from adult tissues.